Good morning, everybody. Thank you very much for staying here connected again. We start with the second seminar, webinar or seminar, online seminar about microbial laboratory. Okay, thank you. Before I start, I want to appreciate the invited speaker had a very interesting presentation this morning with international from USA, from China, and from Chile. Okay, before I start, I want to remember you, we are in the second webinar or Applied Microbial Ecology Laboratory. This is in Spanish, es comprendiendo el papel de las comunidades bacterianas en ecosistemas acuáticos impactados antropogénicamente. That means why did make this presentation in Spanish? Because we had 4 sessions. The 2 first sessions was in Spanish. We collaborate with different speakers during the presentation, but this seminar was focusing more multidisciplinary or transversal audience. Okay, we invited to different people from the Chilean government to the Sun Foundation, and talking about how we protect with river or aquatic ecosystem our region. Okay, we are very appreciate to this person with make participate in this seminar. We started in August 18, and then the second seminar was in September 1st. And then we started 2 new seminars in English, more scientific seminars. We had the invitation. With this webinar was in English. With translate the title is Understanding the Role of Bacterial Community in an Anthropogenic Impacted Aquatic Ecosystem. Okay, in this the first session we had invited speaker to Lin Chang from Beijing Normal University of China, and also Dr. Andy Ogra from University of Florida. Okay, and then was my turn with my introduction about a big project we had with the Japanese government. Okay, and today we had the last session, the 4th session. Is very excited with this presentation. First, we had the Dr. Xiao Rong from Fushu University from China, and the title is Glyphosate Concentration and Bacterial Community Composition in Sediment Sample of Bayandian Lake. And then we had Dr. Kathleen Gray from University of Delaware from USA, and the title is her presentation is the effect of the salinity and biochemical cycling of phosphorus in coastal soil. And finally, we want to show with you, or my group, Dr. Marcos Campos from la Universidad de la Frontera, or La Frontera University. He has the presentation is a spatio-temporal distribution of phosphorus fraction in relation to phosphomosterase activity and bacterial phosphomosterase gene in surface sediment of Booty Lake affected by anthropogenic activity. This is very special lake we had in our region. Okay, not so many study about the bacteria in the lake. Okay, we have continued. I want to say this webinar is organized by our laboratory. We call EMAlab, it's Applied Microbial Ecology Laboratory, and also for scientific and technological bioresearch nucleus, and we call BioRen in our university. But for the support of financial support, we have 3 international cooperation projects. The first is the research network of understanding of the role of bacterial community on organic phosphorus recycling in anthropogenic impacted aquatic ecosystem. This international cooperation is project cooperation we had within La Frontera University and Delaware University and Florida University. We had JC, Dr. JC, like a collaborator, and Andrew Ogram, like a collaborator from Florida. And the second project we had in the lab is the title is Spatial-Temporal Pattern of Bacterial Community in Sediment of Villarrica and Bayadan Lake and their potential as environmental and public health indicator in water resource management policies in the south of Chile and north Chinese Lake. Okay, this international project is its cooperation is with China. We had Dr. Bai and Dr. Cho, which now is presented today about here data or advancing the research of the lake in the China. Okay, then finally we had the third project is a big project we had with the cooperation with Japanese scientists from Dr. Fumito Maruyama from Hiroshima University, and development of harmful algal bloom monitoring method and forecasting system for sustainable aquaculture and coastal fishery in Chile. But all this project is focused in aquatic ecosystem, how to we impacted the aquatic ecosystem, that to make try to make a good collaboration, international collaboration, and good time for this webinar is a good time for share our research and discuss about this topic. I have to say again, this topic is very more scientific, but also we include some before some speaker who talking more social and government issue. We had to be concerned about the sustainability of this aquatic system in Chile. Okay, but we're going to start today, and we had the first what Dr. Xiaorong, but I think we didn't. She's not connected yet. What? That's the reason I will start with Dr. Clancy Gray from University of Delaware, and the title of her presentation is Effect of Salinity on Biochemical Cycle of Phosphorus in Coastal Soil. Okay, I want to check. Please. Sorry.
Hi.
Sorry. Oh, thank you. Ron Xia. Nice to meet you.
Hi. Yeah,
okay.
Nice to meet you too.
Okay, I didn't check you already arrived to this webinar. Oh, thank you very much. I know it's in China. Are you in China, right?
Yes. No, I'm in Fuzhou University. Outside is night, the middle autumn night. Yeah.
Okay.
Today is a middle autumn festival holiday. Today and tomorrow.
Sorry.
Yeah, it's a middle autumn holiday. So
okay.
We wish you all have a happy middle autumn festival. This is a Chinese traditional festival. Yeah.
Oh, nice.
Very big moon, to see moon and eat moon cake. Moon cake.
Okay, we already finished a holiday, big holiday in Chile, the last week. We had Independence Day in Chile. That's a big holiday. We celebrate, make barbecue, folk music in Chile.
Happy time. Okay.
Good time for enjoy.
Yeah.
Okay, please, you can share your presentation. We have started.
Yeah, I click the share link, the share button, but it doesn't react.
But in the button you click, no, didn't work. No,
it doesn't work. Yeah, the button doesn't work. I click the share button, but it doesn't work. So I don't know how to connect.
But we just maybe we get internally we contact by internal chat, private chat, and we start with the Kathleen before, okay? Because we check with Kathleen, Dr. Kathleen from USA. We start first with you, and then we contact you internally to fix the problem. Okay. Okay. That's Kathleen. You can please, Claudio can put the Kathleen in the screen.
All right. So I'm going to do share.
And meanwhile, I will contact with the Real chat. Okay.
Okay. So I'm going to do share screen. I'm going to make sure it's on the right screen. Share. Okay. So presentation. Oh, this is the wrong one. This one. Okay. Yeah. You guys see it okay?
Yes.
Okay, great. Okay. So thank you all for joining us this morning. I'm going to present some of the work I've done over the last 2 years here at the University of Delaware. My primary advisor is Dr. Deb Jassy. I've also done work with Dr. Lisa Stout. We're all in the Department of Plant and Soil Sciences. I'm going to be presenting the effects of salinity on biogeochemical cycling of phosphorus in coastal soil. So sea level rise is a very real threat, particularly in the United States. So if you look here, if you can see my mouse, the places in red are up to 0.3 to 0.4 meter change in sea level rise in 2100. So Delaware is, so here's Cape Cod, this is Massachusetts, this is Long Island. So Delaware is right here in the mid-Atlantic states. So we're expected to see about a 0.2 meter increase in sea level rise over the next century. And this has huge repercussions in terms of saltwater intrusion. So if you look at this figure here, sea level rises, saltwater can infiltrate the freshwater aquifers by the coasts. And this is the same sort of effect that happens whenever there is too much water that's drawn, say due to agricultural processes or just for drinking water, and that can lower the water table. And so both of these have the effect of this increase in influx in saltwater in coastal regions and is quite detrimental to agriculture. Also, this groundwater inundation leads to root decay. So this is not the best scenario. So we've gotten a lot of funding through our National Science Foundation to essentially assess the effect that saltwater intrusion and sea level rise play in the mid-Atlantic states, so specifically Delaware. This is just an image from a location in California. So if you see here, this is due to essentially taking too much fresh water from aquifers. So here, this pole was where the ground level was in 1925, 1955, 1977. So this guy's probably like 2 meters tall. So you're seeing quite a huge drop in where the ground sits because of too much water being taken out. So the same thing happens with sea level rise, as we see with too much water being taken out from aquifers, say to be used for agriculture. It can lead to this saltwater intrusion. So specifically in Delaware Bay, remember this is the mid-Atlantic of the United States. Here, Delaware sits right here. This is New Jersey. And this was a study that was done in 2014. And essentially they saw that with with the seasonal saltwater intrusion, we see a large efflux of methane and carbon dioxide. So it's almost compounding the issue, right? So we have increased sea level rise, we also see an increase in these greenhouse gases. So it's almost like a positive feedback loop, which is not an ideal scenario. So the main point of this study was to determine how this frequency and magnitude of salinity change force particular pathways of nutrient cycling, and what are these specific indicators for such pathways. So we took a coastal soil from Rehoboth Bay, which is just in southern Delaware, and it's ideal because it's largely not impacted by human activity. It's a state park. It's this little, I would really call it a barrier island, but this little strip of land here that has been a state park since the 1960s. So the biggest thing is that we just want an area that was largely untouched by human activity, so we don't have that agriculture influence on this study. So if you note here, we're trying to find a place with just enough phosphate that we could still do the study. It still is significantly lower than what you would find in agricultural soil. It's pretty low nitrogen. But if you note here, the salinity is quite high. It's 29.3 decisiemens per meter. So it is quite true to the name of a tidal salt marsh. And most plants cannot tolerate salinities greater than 4. So we like this area because we really wanted to utilize a group of microbes that were highly salt tolerant for this experimental setup. So what I did is I took wet unaltered sediment and I placed it in these 125 milliliter incubation bottles with phosphate-free Davis minimal medium glucose and then 1 to 3 degrees of salinity. So essentially we have artificial freshwater, mesosaline, and then artificial seawater. And I let them sit over 14 days. If you see here, I also have these larger bottles with 120 grams of sediment and then 150 milliliters of medium. So essentially double what I had in these bottles. And that was because I was trying to sample the headspace, so the gases that these microbes are giving off over the course of the experiment. And of course, I autoclaved the control groups before I added the medium. So I sampled them over 1, 2, 3, 4, 6, 10, and 14 days. Separated them through centrifugation. I took the sediment and supernatant and immediately froze them at -C and then freeze dried them. And that was what I used for all these analyses. So the data that we were looking for is the phosphorus concentrations, different pools, nitrogen isotopes, carbon isotopes, and then nitrate and ammonium concentrations of these different parts. So of the soil, of the supernatant or dissolve phase, and then of the headspace. In terms of microbial parameters, we sent off raw soil to Dr. Thomas Hansen's lab here at the University of Delaware, so we can get essentially microbiome assemblage data. Then we had dehydrogenase activity and alkaline phosphatase activity done by Lisa Stout. And then the gene expressions were done by Marco. Sorry, I should have put down Marco because he did it directly at the Frontier University in Chile. So for dehydrogenase activity, these are the different treatments. So seawater, mesohaline, and freshwater. And if you note here, it increases quite a bit by this day 14. So just keep that in mind when we look at the phosphate pool data. Same with alkaline phosphatase. So this is essentially saying that if cells if microbes are phosphate limited, so normally cells will just take pure phosphate, so PO43 minus. But if there's not enough dissolved inorganic phosphate, so it's PO43 minus, they will cleave like monoesters, diesters. So essentially these organic phosphorus using alkaline phosphatase. So by cleaving it, they're able to turn that organic phosphorus to a usable form. So to that dissolved inorganic phosphate, that PO4 is what they can readily use. And so if you note here, by this day 14, this mesosaline sample has quite a large increase in alkaline phosphatase. It could just be because it's used up all of the easily accessible DIP, so dissolved inorganic phosphate, first. And that's why its activity increased by day 14. All right, so now sort of into the nitty-gritty of carbon and nitrogen isotopes. So our starting sediment had a per mil value about -20 per mil. And so what I want you guys to think about is just the relative, these excursions of like a 1 per mil or increase or decrease of per mil values over time. So essentially we're looking at relative values. So what we see is for this freshwater sample, it's the most depleted compared to these other two samples. And then they stabilize over time. So what we think might just have occurred is that there's just a resuspension of organics in that supernatant, so that liquid portion over the first few days, which might cause this signal. And then it drops by day 14. And so what can also cause a change in this delta 13C is essentially just utilization of carbon species. So dissolved organic carbon and dissolved inorganic carbon. We did not differentiate between those two different forms of carbon. This is bulk carbon isotopes. But what I would assume happened, for especially this freshwater sample, is that it was probably utilizing more of the dissolved organic carbon species. So whereas compared to the seawater and the mesosaline sample, we don't really see that change from that initial value, which is kind of sort of telling us that those microbes, at least in the freshwater sample, they're probably having to change the metabolic processes just slightly in order to use carbon for their life species. Okay, so for the control group, this is really interesting in that we see for seawater, versus freshwater mesosaline, it almost shadows each other. And so we see this freshwater control was essentially became the most depleted over time, while this mesosaline control was only slightly depleted, and the seawater control became slightly enriched compared to the original sediment. So my inclination as to why we're seeing this inverse in signal, remember this is our control group, so there should be little to no microbial activity because it was autoclaved. And so what I'm assuming happened here is that the cations that were present in that seawater could be interacting with the dissolved inorganic carbon species. And that's why we're seeing this change in our delta 13C values. Is essentially there's this interaction between the way that I made this artificial seawater and the sediment itself that was abiotic. So there's no microbes involved. For samples for nitrogen isotopes, so this I'm still thinking about because there in this site and also in most tidal salt marshes, there's usually high initial ammonium concentrations. And so we think about denitrification in general. You're changing nitrate to nitrite to ammonia. And I'm still waiting on that data, that concentration data, to see how over the course of the experiment, those different concentrations have changed over time. But what I'm assuming happens, again, I don't have that data yet, is with this initial decrease is that we're perhaps seeing denitrification happening in these samples. So we'll have to see how that data turns out. So for the control group, we interestingly also see this dip here. So we'll just have to see how that concentration data pans out. Because again, this should all be abiotically induced. All right, so for phosphate, phosphate is unique in that it interacts with different elements in different ways. And when it comes to oceanic sediment, we usually use this process called SEDEX in order to sequentially extract those different phosphate pools, whether it be iron and aluminum oxides or quote unquote or organic phosphorus pool. But for soil, we do this sequential headley extraction. So you essentially add a different, not necessarily like a reagent, but a different chemical in order to get that specific pool. And different pools mean different things. So for water, that's literally you just put it in water for 2 hours, you shake it, and that's in the supernatant is our water pool. And that is extremely loosely sorbed P. It's very bioavailable. The second set is this sodium bicarbonate pool. Same thing, is extremely bioavailable. The signals that we get from the sodium bicarbonate pool are usually much more clear than this water pool. And then continuing on for the next sequential extraction, we use a strong base, so sodium hydroxide, in order to look at our iron and aluminum oxide bound P. This is less bioavailable. So essentially, when microbes only try getting into these pools once they use up all the phosphorus in these first two pools. And then this last pool is this nitric acid pool, essentially calcium phosphate. That includes stuff like, so a very good example of calcium phosphate is just like your teeth, it's apatite. So like bones, teeth, etc. And to a lesser extent, calcium carbonate can interact with phosphorus to make that calcium phosphorus compound. But both of these are less bioavailable. So for this water pool, again, this data is a little messy because as that super bioavailable pool, it's very, very loosely sorbed. We can't really say much about what's going on using just this. So I'm going to just kind of zoom ahead to this the sodium bicarbonate pool, which will tell us a lot more information just because it's a little bit more constrained to the sediment itself. So in blue here, here we have our samples, and then in the warmer colors, we have our control groups. So if you just look here at the freshwater sample, you'll see over time it's not really changing much, if at all, from those initial concentrations, which are, to remind you again, they're quite low. So for like agricultural soils, these might be as high as like 10 to 50 micromole per gram. But this site's quite low because again, we wanted a site that was as close to like non-anthropogenically influenced concentrations as possible. But if we see here at freshwater sample, it's not really using a lot of this bioavailable P pool, and it could just be, or what I'm assuming is those microbes just, they're not really well adapted to pure freshwater because it's a tidal marsh. So they're much more inclined to be okay with, they're much more salt tolerant. Which if we see here for the seawater sample, we see there's really not a decrease in concentrations. There's an increase, and then they decrease as they use it up. And then what I'm assuming at this, that's day 14, is that a lot of the bioavailable P has been used up, and microbes are dying off because they've been in those little bottles for 2 weeks straight, which you kind of see with that DHA activity. So let's see what else. Okay, I'm gonna go to the next slide. So if you just remember here, these two treatments are corresponding to calcium phosphate, which is this nitric acid, and then sodium hydroxide is this iron oxide P and aluminum oxide P. A lot of previous studies have found that as salinity increases, dissolved inorganic phosphate also increases. So this decrease in the nitric acid pool, specifically in this control group over the first few days, indicates pre-release from this nitric acid pool might be transferring to the sodium hydroxide pool, which is really cool that we see that. But we definitely see that increase in that sodium hydroxide pool over the course of this experiment. And it just goes back to, with an increase in salinity, we're going to see more of those iron oxides and aluminum oxides being released. And this is going to be true whether or not you have microbes in those incubation bottles or not, which is why we see also the control groups doing the same thing. So this increased salinity releases cation species, which affects phosphorus availability, as that phosphorus could have previously been bound to calcium, iron, or aluminum. And then that released phosphorus can in turn be resorbed to calcite or other cations, or used by microorganisms. So this is data that I just got from Marco Campos about 2 months ago. So this is just gene activity here. So if you take a look at the phospho-D and phospho-X genes, we see there's actually quite a bit of those genes present in this mesosaline treatment, which surprises me because I would have thought that we would see a higher presence in the pure seawater treatment. But it could be that that salinity might have been even too high for those microbes themselves. And we are waiting on, again, that microbial microassemblage data, which will just better elucidate what microbes are present. It could just be that those microbes, it was just too salty for them, which is why we don't see as high of a presence of those phospho-X and phospho-D genes in that seawater treatment, whereas you see it quite a bit higher in that mesosaline treatment. So we're thinking that maybe they're adapting to salt stress. Again, we're not completely sure. We're still thinking about that. This result is consistent with that increase in that sodium bicarbonate and sodium hydroxide pools which showed a higher increase after 14 days of incubation. And then if you note here, this is a gene that essentially corresponds to that denitrification. So same thing as that we're seeing higher amount of that gene present in that mesosaline group. It'll be interesting to see what our nitrate and ammonium concentration data says. So my hunch is that we're going to be seeing higher concentrations of ammonia at the start of this experiment. It's going to maybe initially decrease, but then it might be high again by day 14. I definitely do think that we're going to see nitrates probably decreasing over the course of the experiment. But again, we're waiting on that data. So we'll see how that works out. So just some conclusions here. So that natural resuspension of organics in the supernatant may have caused that changing delta 15C and delta 15N. We're assuming that the supernatant is heavier and delta 13C for mass balance. Our freshwater treatment shows the largest change in delta 15N, which is enrichment. It also shows the largest depletion in delta 13C. As I mentioned before, that delta 15N could be due to ammonium nitrate conversion, so nitrification. We could also be seeing denitrification present in these microbes, which again, we're waiting on that data. Let's see. So just another recap. So as salinity increases, we know that that dissolves, so that inorganic phosphates also increases. Our decrease in the nitric acid P, so calcium P, an increase in iron and aluminum oxide pools over time. There'll be P released from that calcium P is being absorbed to the iron and aluminum oxide P. The sodium bicarbonate pools, remember that's our readily bioavailable phosphorus pool, and samples and controls to show opposite trends. So our decrease in sodium bicarbonate pools over time show our microbes are using more bioavailable P. Again, this increase in that sodium bicarbonate pool in the seawater and mesosaline treatments at day 14, it corresponds to a minimum in DHA activity. So I'm just assuming those microbes essentially were dying off by day 14. So it's a signal that there's a rebound in bioavailable P due to decrease in microbial activity. And then among those three salinity levels tested, the expression of APA was highest in mesosaline seawater treatments. So I'm assuming that these microorganisms, they're adapted to deal with salt stress, and they can increase their activity to cycle P pools to meet their metabolic and nutritional demands. But just looking at the gene expression data, I have a feeling that there's something unique happening with that mesosaline treatment that we're not really seeing in the seawater treatment. So I'll be interested to see what the microbial assemblage data says along with the nitrate concentration data. So there's still a few things that I still need to do. So we do want to do a mass balance. So we want to measure that supernatant, that liquid part, for delta of water, delta 15N, and delta 13C. Unfortunately, our Flash EA was having some issues the last few weeks, but it should be hopefully online maybe by the end of this week. And then also measure headspace gases for delta 15N and delta 13C. And again, we're waiting on that microbiome core analysis by Thomas Hansen at the University of Delaware. All right, and then I guess I will take a few questions if you guys have questions. I have there's questions in the chat.
Thank you very much, Caroline. But I have to remember to the audience, you can write with social media your question, but now we can see here in the system. We don't have question from the audience yet, but I had some question. You had more different with fresh water compared to seawater. What is the difference with do you have some data about the difference in the microbiome within both in fresh water or seawater? Is total different, the activity or the functionality or the taxas, the structure there?
Yeah, were you asking if I know what taxa are present.
Yes, not only taxa, only the functionality. In the previous work, I don't know, there's some historical work in the different with the fresh water and seawater in your region.
If like any other work has been done in this area concerning the microbes, no. There hasn't been. So the biggest push for using this field site is that there's a bunch of different people at UD working on it, and so there's another group that's doing essentially looking at how iron and sulfur, the role that those elements play in microbial cycling of the same area. But same thing is that we don't know anything about the microbes, the microbial assemblage just yet. And so we sit in all this raw soil data. It was like months ago. So we're all waiting on the same thing. Yeah. So there's been a lot of work done both north and south of this field site. But in terms of the field site itself, we don't specifically know what's living there just yet. So yeah, I'm just like being very anxious. I keep like asking Thomas Hansen, like, have you gotten the data yet? So I don't know what's taking forever. But yeah, as far as I know, no one knows exactly what's living there. And again, I'm not a microbiologist, so I wouldn't know off the top of my head. Sure. But yeah, I'm interested to know too, just to see what's going on, because it goes back to that that mesosaline treatment is doing something unique, especially as with the gene expression data. We're seeing more of it than we are seeing in the seawater treatment data. So yeah, I'm assuming it probably has to do with the fact that the Delaware Bay itself, it's tidally influenced, so it does see salinity fluctuations just over the course of the day. And so maybe that's what's going on. It's just the microbes do best at that mesosaline salinity.
Okay, we had very interesting with the nutrient availability increase the some microorganism bloom, the fast growth.
Yeah.
But maybe interesting to how to this salinity related to this some bloom, algal bloom or cyanobacterial bloom.
Oh yeah, that's a really good point. And so we didn't spike any of this treatment with phosphorus, but it would be interesting to see if we spiked it with nitrogen and phosphorus to see how it affected their growth and then how salinity affected that. It would make a lot of these analyses a lot easier if there is more phosphorus and nitrogen available. But yeah, that's a really good point. I'm not exactly sure the role that salinity does play in just those algal blooms, because I was always just assume that they're just if there's a surplus nitrogen or phosphorus, like we'll see a lot more of this algae. But I'm curious if it's more saline, we won't see as much growth, or if it's more freshwater, we'll see less growth. So yeah, that's a good point. I'm actually not sure.
Yes, because last week we had a presentation from Audiogram. He make a cenote study with cenote microbiome. This is in Mexico. Dr. had a lot of experience in the Everglades in Florida. That he did a study about the structure, the community, the microbiome there, and with a similar function, but the taxas is different. Like the nitrogen cycle, that the same functionality the system in nitrogen cycling, but the taxa is totally different to the cenote to the another in the case the Everglades. That's I think maybe we can make some different. Maybe it's working the nitrogen cycling in that system, but the taxa involved in this process is maybe the same or different, or they're influenced by salinity too. That's very interesting. Thank you very much. I think we are on time for continue the next one. I also remember to the audience want to make some send out your question, and we had time to finish the self presentation. We continue talking about this one. Okay, if someone had another question for Kathleen, please write and send to us, and we can ask her later. Okay. Okay. Thank you, Kathleen.
All right. Thanks.
Okay. Now we continue with the Dr. Xiao.
Hi, Mirko. Hi, everyone.
Hello.
Yeah, nice to meet you. I'm sorry I'm late. I'm late. done up to this online webinar, and I just gave a brief introduction of our work in the Baiyangdian Lake. Yeah, we just because based on this China joint program on the water resources management, the financial support and the cooperation with Dr. Bai of Beijing Normal University, we collected the sediment samples. First of all, I want to introduce the Baiyangdian Lake. The Baiyangdian Lake is called as the pearl of northern China. Can you see my screen?
Not yet. Please share it, please. Okay, now it's opening. Okay, now.
Yeah,
we are ready.
Yeah.
Okay, go ahead.
I share the screen.
Yes.
Yeah. Do
okay,
you?
Perfect.
Yeah. Okay. Yeah. And the Baiyangdian Lake is called the pearl of northern China, and it's the largest lake in Haihe Basin. And this is where the lake located in the north of China in Hebei province, and within the Baoding city. Here is the shape of the lake. And the lake has an area of 366 square kilometers. It shrank from the over 500 square kilometers in 1950s. So the area is decreasing because of the reclamation, and the water depth is 1 meter to 6 meter because of the human activities, anthropogenic threats, such as the population increase, the reclamation, the agricultural industry, and urban pollution into the lake. So there are many environmental problems coming out, such as terrestrialization, water storage decrease, eutrophication, contamination, water quality deterioration, and biodiversity decline. So many studies have checked the heavy metal pollution, POPs pollution, and the eutrophication problems in this lake. Since the herbicide glyphosate used since 1970s, for the wide weight controlling, but there is little information about the glyphosate accumulation in the lake sediment. So based on the China joint project, Dr. Milk suggests us to check the herbicide pollution in the lake. So we start to check the glyphosate accumulation in the sediment. We collected the sediment samples since the October last year. We collected 56 sediment samples, cover throughout the whole lake. And here is I cut the map from the Google Earth. This is the lake area, the dark green color, and around the lake distributed a lot of cities, towns, agriculture land, and also there are many villages within the lakes around the lakeside. So the lake area is decreasing, and the water quality is declining. The results of the glyphosate accumulation in the sediment, we checked the 3 seasons. One is October of last year. The detection in 56 sediment samples, 11 sediment samples were found to contain the detectable glyphosate concentration. So the detection frequency is 20%. The concentration ranged from 0 up to 4 milligram per kilogram, with a mean value of 0.9 milligram per kilogram. This is the distribution of glyphosate in the sediment throughout the lake. We can see the glyphosate concentrate in the northwest and northeast because of the river input into the lake and the output of the lake. In the April of this year, we collected 48 sediment samples, and 22 sediment samples were found to contain detectable glyphosate concentration up to 4.38 milligram per kilogram, with a mean concentration of 0.4 milligram per kilogram. And the hot pot is located at the southeast of the lake. Here is the reed field. The local people plant reed for the reed roof, reed straw mat, and reed blanket production, and reed production for paper production. And this August, we also collected 48 samples in some typical zones. We found the glyphosate concentration was up to 8.6 milligram per kilogram, with the mean value is 3.9 milligram per kilogram. However, despite the detection frequency is just 13%. So from the glyphosate concentration distribution pattern, we can see the special distribution pattern. This distribution has the different patterns in different places, in different part of the lake. And the hot pot distribute in the northwest, northeast, and the southeast, and the center of the lake, or the upper part. According to the soil environmental quality standard in China, the threshold of glyphosate in soil is 0.5 milligram per kilogram, above the value indicating the possibility of pollution hazard. So we can see the glyphosate concentration in Baiyangdian Lake is exceeding the threshold of glyphosate according to the soil and environmental quality standard, which is over 0.5 milligram per kilogram. Yeah, that the mean value is over 3 milligram per kilogram, or in August. In April is 0.4 milligram per kilogram. In October, 0.9 milligram per kilogram. So the glyphosate accumulation in sediment is worthy being noticed in the Baiyangdian Lake. And we compared the data with the studies in most studied glyphosate polluted area at the pampas of Argentina. We see the pampas showed the first order strain in the agriculture area contained the glyphosate concentration above 2 milligram per kilogram, and the range is 0.1 to 5 milligram per kilogram. So the glyphosate concentration in Baiyangdian Lake is near the stream sediment of pampas in Argentina, where the glyphosate concentration is usually regarded as the high level all around the world. So the glyphosate pollution in Baiyangdian may be a serious problem. We need to further study it. And then we used the risk quotient RQ index and the toxic unit TU index to assess the chronic risk and the toxic unit of glyphosate to sediment dwelling organisms and aquatic biota in the Baiyangdian Lake. We see the RQ value. Here is the explanation for the RQ value and the TU value. The RQ value above 1 indicate high chronic risk, and the TU above 1 indicates acute risk. We can see in October last year, the RQ of Zaobeikou town and Fuhe River. Zaobeikou town is located in the northeast, the hot pot of the October side, and the Fuhe River. Fuhe River is northwest. The inflow into the lake, the river inflow into the lake has, and the Zaodadian artificial ditch, which connected the Fuhe River into the lake. So the three sites should the RQ value exceeding the value 1, indicated the high chronic risk of the glyphosate to sediment dwelling organisms. In August of this year, the Caiputai village is located in the southeast corner, where the local people planted the reed filmed for roof, blanket, or mat production. Here, the RQ is above 1. And Nanliuzhuang village, which is located within the lake, in the north part of the lake, next to the Anxin city, and the Chuantou village, which is located in the center of the lake, and is a water village. The household waste, living waste, all were discharged into the lake. And also the villages next to the city receiving a lot of waste from the city. So the RQ values should high value. Here the toxic unit in Nanliuzhuang village and the Chuantou village also showed the values exceeding 1. That means the Nanliuzhuang village and the Chuantou village has the highest values of ecological risk due to the glyphosate accumulation in the sediment. So they may produce the risk to sediment dwelling organisms and the aquatic biota. Compared with the values in the pampas of Argentina, the TU values and the RQ values is similar to those of pampas Argentina. 0.
Hello, I think we had some problem with the connection. We wait for a few minutes to if Dr. Xiao can connect again. Sorry for this problem.
Give more attention in this area, so we need to we need to pay more attention to the risk of the glyphosate accumulation in the sediments in the Bayandian, especially in the sites next to the city and
Sorry again, we had a problem with the connection with Dr. Xiao. We wait for a few minutes for reconnect here. We are waiting for the reconnection of Dr. Xiao. He's showing her result about this glyphosate content in the lake sediment in China. Okay, we wait. She can connect again. I tried to contact her by chat. Is ready there. We are waiting for the answer from Dr. Xiao. The manager informed me she's already disconnected for this session, that we wait again. She's connected now. Sorry.
Yeah,
sorry.
yeah.
We have problem with your connection.
Sorry, Milk. The link was cut down. Sorry for some mistakes.
Go back.
Yeah. Can I continue?
Yes. Sure.
Yeah. Okay. Sorry. So here. Can you see my screen?
No, now it's opening.
Can you see my?
Okay. Yeah, but you can go back for 2 slides. Please go back, please.
Which slide?
Okay, continue. When you're talking about the Pampa Argentine, okay, Pampa Argentine.
Okay.
More.
Okay. This one here. Okay. Yes, in the August we find the RQ in Taiputai village, where the local people plant reed for the mat production or the roof or the the paper production for the reed plant. Here and the Tranto village within the lake have very high RQ value. And in August, the toxic unit in the Nanliuzhuang village next to the Anxin city receive a lot of the waste from the city, and the Tranto village, the Nanliuzhuang village, Tranto village, both have high RQ and toxic unit here. So we compared the RQ and the TU values with those of Pampas and Jitila. We found the Bayandian Lake showed the high risk due to the glyphosate accumulation in the sediment, comparable to those of the sediment from Pampas and Jitila. They also presented up to 5 toxic units, up to 5, and RQ up to 6. And also here in our study, the RQ value up to over 6, and the TU value over 1. So that means the the glyphosate may produce high chronic risk and high toxic risk, the acute risk to the sediment dwelling organisms and aquatic biota. And here is the Nanyuozhuang village next to the city. So receive a lot of garbage from and waste from the city. There is a high value of RQ, risk quotient, and the toxic unit values. And here is the Chuantou Lake village, the water village within the lake. A lot of waste from the local people living residents, household waste were discharged directly into the lake. The water quality there is bad and it's smelling. When we go there to collect the samples, we smell the water smelling. So we checked the bacterial community using the high throughput sequence of 16 RNA genes on Illumina MISIC platform, and we divided our sediments into 3 groups, which containing high glyphosate concentration above 2 milligram per kilogram, medium glyphosate concentration 0.5 to 2 milligram per kilogram, and no glyphosate concentration less than 0.5 milligram per kilogram. So we divided our sediment samples into 3 groups based on the glyphosate concentration. From the PCOA analyze, we can see the samples from the glyphosate highly accumulated sediments clustered into one group, and the medium and low group, the samples clustered, and they have the overlap. And based on the bacterial community, the phenon difference, we can see for the phyla of Proteobacteria, the dark green color of Proteobacteria, and the Campylobacteriota here, the light green part, showed relative high abundance in the glyphosate high group, while the chloroflaxi and the Actylobacteriota and Acidobacteriota and the Firmicutes showed relatively lower abundance in the glyphosate high group. Furthermore, we find some typical operational taxonomic unit, the OTU, the very typical, different in the three glyphosate accumulation level sediments. The significant difference were found in the OTU abundance in the different groups of sediment. And then we check the correlations between the abundance of typical bacteria OTU, the dominant phenon, Proteobacteria, the Campylobacteriota and the Actylobacteriota, Acidobacteriota, Firmicutes, Chloroflex, and the sensitive OTU were found, especially for the Proteobacteria and the Campylobacteriota showed the significant positive correlations with glyphosate concentration, while the OTU belonging to Actylobacteriota, Acidobacteriota, Firmicutes, Chloroflex showed significant negative correlations with glyphosate concentration. and we analyze the network of OTU, typical OTU, considering their interaction with glyphosate and other environmental factors. The red lines mean significant positive, green lines mean significant negative correlations. We can see the Proteobacteria, the blue circle, showed positive correlations within the OTU, benign to the Proteobacteria. And the positive correlations were found also within the inhibited bacteria, so-called temporarily, the Chlorophyllaceae and the Actinobacteriota. But the negative correlations were found between the so-called stimulated bacteria, Proteobacteria, and the inhibited bacteria, Actinobacteria, and the Chlorophyllaceae, the Firmicutes, and the light green, the Campylobacteria. Campylobacteria showed positive correlations with Proteobacteria. So we just found the Proteobacteria showed positive correlations with the Campylobacteriota, and also the positive correlations were found between the Chlorophyllax and the Actinobacteriota, but the negative correlations were found between the Proteobacteria and the Actinobacteriota. Further, we found in the right finger, the Proteobacteria showed positive correlations with glyphosate concentration, while the Firmicutes and the Chlorophyllaceae and the Acidobacteriota and Actinobacteriota showed negative correlations with glyphosate accumulation in sediment. Also, the environmental factors such as the electric conductivity, the pH, the nitrate content in the sediment also impacted the OTU, the bacteria abundance in the sediment. According to the publications which studied the potential functions of the typical bacteria, we found the stimulated bacteria by glyphosate in this study. Some typical OTU were related with the sulfur oxidizing bacteria and the deleteriferous. Also, related to the reduction of nitrate. But the inhibited bacteria by glyphosate in this study, some typical OTU were related with the metabolism of some refractory organics, the degradation of acetic acid, denitrification, nitrogen removal, and some organics decomposition, such as the Chlorophyllaceae was reported to connected with the plant detritus decomposition and refractory dissolved organic matter biodegradation and methane metabolism. Furthermore, we use the FAPROTEX, the functional annotation of prokaryotic taxa, the database, to analyze the bacterial metabolic functions in high, medium, and low glyphosate concentrated sediments. In this study, we found the nitrogen respiration in the nitrogen respiration, the nitrate respiration, and chlorate reduces and the sulfite respiration were stimulated by glyphosate, which is significantly higher in the high group glyphosate highly accumulated sediments.
Sorry, we had again
Yeah.
this. Okay.
We cannot hear.
Just few minutes.
Yeah.
You can explain again the
Yeah, we take the median and the null group
Mm.
into one group. We check the high the glyphosate highly accumulated sediments should the inhibited manganese oxidation and inhibited nitrate respiration, inhibited nitrate denitrification, but the stimulated intracellular parasites and human pathogens, nosocomial, sulfite respiration in the high glyphosate, highly accumulated sediments. So we can, from the information from the published documents and our function predictions by FARPROTEX, we can see some bacterial functions were stimulated by the glyphosate accumulation, but some bacterial function were inhibited by the glyphosate accumulation. There are some findings. We find the glyphosate showed a medium to high accumulation level in Bayandian Lake sediment and the chronic risk to sediment dwelling organisms despite the detection frequency was less than 50%. Significantly higher relative abundance of Proteobacteria and Campylobacteriota, while significantly lower relative abundance of Actinobacteriota, Acidobacteriota, Firmicutes, and Chloroflexi were found in sediment bacterial communities from glyphosate high group, the medium and low group. Network analysis emphasized the significantly positive correlations with stimulated bacteria and with inhibited bacteria, respectively, and between glyphosate and stimulated bacteria, while negative correlations between stimulated and inhibited bacteria, as well as between glyphosate and the inhibited bacteria. Also, the glyphosate-stimulated bacteria were mainly identified as sulfur oxidizing bacteria and nitrate-reducing bacteria, while glyphosate-inhibited bacteria were mainly identified as oxalate-degrading bacteria and denitrifying bacteria. Bacterial community function predicted by FARPROTEX confirmed the stimulated bacterial metabolic functions of sulfite respiration, Dacarin oxidation, and nitrate reduction, but functions of fermentation, manganese oxidation, photoheterotrophy, and nitrite denitrification were suppressed in sediments with high glyphosate concentration. Glyphosate concentration and its interactions with bacteria were strongly affected by sediment pH, electric conductivity, dissolved organic carbon, and nitrate content. So these are some findings. Our work was financially supported by National Science Foundation of China for the China-Chile Joint Projects on Water Resources Management. We gratefully acknowledge Professors Bai Junhong, Andrew Ogren, Miyoko, Zhang Kegang, and lab members Kuang Bo, Wang Shen, Zhang Ling, Wei Zuoxun, Liang Jinfeng, Wang Yaqi, Zhang Guangliang, and other anonymous colleges. So today and tomorrow is our Chinese traditional festivals. Wish you all have a happy Mid-Autumn Festival. Thank you. That's my presentation. If you have any questions, so please don't hesitate to ask or give us very important suggestions. Thank you.
Okay.
Thank you, Milk.
Thank you very much, Joe. We had time for one question, but remember, in the end of the third presentation, we have more time for questions. But we are on time for the next presentation. I'll only ask only one question for our audience.
Sorry. Yeah, the natural.
Yes, the question. Can you hear me? Can you hear me? But I will read a question from Dr. Fumito Maruyama. And this question is the figure number 4 network analysis based on Pearson or Spearman? Or how many sampling were included in this analysis? And how is the statistical reliability? Okay, you can read this question.
Sorry, the internet is not so influent, and countdown sometimes not connected, sometimes connected. So yes, I just listen and little information. We collected 56 sediment samples in October last year, and this April and August of this year we collected 48 sediment samples, and we analyzed the bacterial community using 15 typical sediment samples.
Okay, and about the analysis, Pearson or Spearman? The network analysis. This is based on Pearson and Spearman. Maybe we can show the question again. Please read your screen.
Okay. Which slide?
What question? What more detail about your analysis? It's a Pearson or Spearman. You already said the number of the sample. And
is the figure 4 network analyzed based on Pearson figure 4 here, figure 4. Based on Spearman, how many samples were included in this analysis? 15 samples. Yeah. How is the statistic reliability? Just the link showed the significant, the red line or the green line, both showed the significant correlations based on the Spearman correlation. The p-value above 0.5.
Okay, 0.05. Okay.
Yeah.
Thank you. Thank you.
Thank you, Mirko.
Okay, we continue with the SARS presentation. Now we invite to Dr. Marcos Campo. Marcos
everyone.
Campo is a colleague from my laboratory. He works now in this time. Dr. Marcos shows some data from Buddy Lake. Okay, Buddy Lake. Sorry, they usually say in English. Buddy Lake. Okay, please, Marco. Please share your screen, and we continue with your presentation.
Okay. First of all, thanks very much for the invitation. Thanks everyone, the researchers here, because it's a good opportunity to share different data about our projects. And also I have been involved in the international cooperation with China in this project, and also with the REDES project to working with the USA. So it's very good for us, meet us face to face, see how we can continue doing next projects or next objectives, and assess new experiments. So it's good for me, good for all all of us. In this order, I present part of my my researching, postdoctoral research, and I share. I don't know if you can see my screen. Can you see my screen? Yes, now it's okay. This research was assessing the researching about what about the status of bacterial community in rivers. In a previous webinar from the past year, I present result about the rivers, Imperial and Tolten River, the two main rivers here in our region. And now I present part of this research about the Buddy Lake and how the distribution of phosphorus fraction and relation with the phosphomonosterase activity and bacterial phosphomonosterase genes has a spatiotemporal distribution in surface sediments of Buddy Lake, a very special lake here in our region, or even at the continental level. The main reason to study this is because threatened by high anthropogenic activities. So it's a good opportunity to know what is happening there with the status of the microbial communities. We know that the phosphorus is a very important element in life, is taking part of biological molecules such as DNA, phospholipids, so it's very important for replication of cells, for the construction of bones in our organisms. So microorganisms are also the life had to take this element from the nature, and sometimes this element is under the organic form. The unique form that is usually taken by microorganisms is by the inorganic form. So they have to assess this molecule by hydrolyzing this molecule with different kind of enzymes, where, for example, phosphomonosterase are very important to mineralizing this molecule to obtain orthophosphate. That is very important for this molecule. What is the problem with the phosphorus in shallow lakes or lotic and lentic ecosystems? That the phosphorus at the stable levels is good for the productivity of lakes. But what happens when the inputs are too huge? Is leading to the eutrophication. And the eutrophication is that when the microorganisms that has a faster replication, such as phytoplankton or certain bacteria, are taking this element that is scarce in commonly, are taken to with the water of this environment. And why is important phosphorus? Because phosphorus is recycling in sediments, and there is accumulate and stabilize with the iron or cations there, but the fluctuations in the abiotic and biotic conditions can towards this organic molecule to inorganic molecule, inorganic forms. By this, microorganisms can take this molecule and fast grow, and let's from, for example, from a mesotropic or oligotrophic environment to eutrophic environment. That is one of the most concerning issues worldwide in recent years. What about the bacteria? Why is important to study bacteria? This is because bacteria has widespread enzymes such as alkaline phosphomonosterase and as acid phosphomonosterase that is assessing this organic molecule, mineralizing this to take orthophosphate. So we can study the status of bacterial community, assessing the activity of these enzymes, or also by the quantificating the abundance of genes. Because, for example, alkaline phosphatase is encoded in aquatic environment, also in soils, by phosphomonosterase FOD and FOX genes. We have these two allotypes. And for acidic, for the acid phosphomonosterase, we have the FOC genes. So assessing these genes, we can see how the bacterial community is taking part of this process in the recycling of phosphorus there in sediments. Why the study Buddy Lake? Buddy Lake is a coastal lake, is a coastal shallow lake, also is brackish. This lake has a brackish waters, salty water, and it's close to the sea, has this particular form. It's a very important environment to the surrounding settlements, because there are living a big population of Mapuche, Lafkenche people. That is a ethnic population in our country. Buddy Lake is providing several ecosystem services for these people, also for sustaining the regional activities. If you can see there, people is very, very recognized for they construct buildings and the activities such as per years, many years ago. And there we can also find different kind of species, fishes that are already unique there in this environment, or this also harbor, for example, the black-necked swan. That is a species of swan very protected in our country. This among others species that are very unique there. And how you can see there, Buddy Lake is considered one of the few salty lagoons in South America. So for sure we have there a unique environment that we have to protect. But what about is concerning to study the status of microbiological activity there? Because if even we have to protect this environment, is actually or currently is not being protected, because there are very high anthropogenic activities such as cattle, livestock, the agriculture. Also there is very common to find this kind of agriculture where erode soil particles are reaching the water and transporting organic phosphorus and other nutrients or organic matter also there. For forestry, that there is a very, very intensity with exogenous species such as eucalyptus or pinus. But this is the problem, because the people need these activities, but these activities also are threatening the quality of Buddy Lake. For this reason, we decided to study this lake, because we need to push for new studies focusing on the microbial community, trying to develop or trying to see what is happening with the ecological roles of microbial there, and try to support new policies for the protection of this environment. In this reason, at the light of this, we decide determine the distribution of P species in sediment of this lake, analyze the PACE activity and PACE bacterial gene abundance. For this research, we collect some superficial sediments, two planes. The first one is a spatial plane where we identify two sampling points with less anthropogenized activity. These were Paucho site and Comue stream site. that are not too threatened by anthropogenic activities, but also we include 3 different points with or we characterize this as more anthropogizing sites that were the mode of the Temo stream that is contributing with a lot of sediments, material from agricultural lands, and Puerto Dominguez sites that is just in front of the biggest human settlement that is with the same name, Puerto Dominguez, and Temo site that is quite similar to the Temo stream site. We took the sediment with a Peterson-like grab and by triplicates in 2 different seasons. These were winter and summer. And we also later of this, from this sediment, we took different aliquots for the pace activity and pace quantification of genes. Also, the chemical study of the P status and other chemical analysis. The P species and chemical analysis were the first one attempt were by a P fractionation that was using the Ivanov method, that is a sequential extraction, starting with a sodium carbonate to take the libal fraction, the inorganic and the organic fraction. Also, the same extractant were used for the microbial estimation after the fumigation of the sample, and the modeled libel fraction was obtained with hydrochloride acid, and the moderate organic fraction was obtained with the sodium hydroxidum. You see where we estimate the fraction bound to the fulvic acid, and the non-libal fraction was estimated also with this extractant that is bound to the humic acid fraction, the residual fraction. And the total inorganic phosphorus was estimated at the addition of this fraction in red, and the total organic fraction was the estimation of the green ones. And the total phosphorus of all the sediment, all kind of species there, was the addition of each one of these this fraction. Also, by working with Anthony Hollenbach from the Dr. Dev Yaisin in USA, from our university, we can study also the 31P NMR analysis. There were detected different species of organic and inorganic phosphorus forms, where the main were orthophosphate, pyrophosphate as the inorganic forms, and there the phosphomonoesters and phosphodiesters, such as DNA and non-DNA diesters, were the main peaks obtained in the NMR spectra. Also estimate the total carbon and the total nitrogen. And the pace activity was estimated with the Tabatay and Bremer method, using paranitrophenyl phosphate as a substrate, and the assessing of the qPCR for the abundance of the total bacteria community by the 16S RNA, and the PACE genes were using this method described for these authors. And also the total abundance, the absolute abundance, were relativized in function to the 16S RNA gene to have a better estimation of the quantity of these genes. All of these parameters were analyzed in the PCA analysis, and the physical-chemical related parameters also were subject to ANOVA parametric tests and Pearson correlations analysis to see what are the correlation there with between or among the parameters. About the PEP fractionation, the main results were that the spatial plane sediments from the more anthropogenic sampling site contain significantly higher levels of libel, microbial, moderately non-libel, and the total organic and inorganic phosphorus, and also the total phosphorus fraction than those from the less anthropogenic sites. These results were quite similar to results that have been obtained from other lakes with elevated anthropogenic intervention and troublesome with the eutrophic status. So our results at this level are very well correlated with this information. At the temporal plane, the label P and the microbial P showed a higher significant levels in summer than in winter. Conversely, the moderately libel and non-libel P, and also the total organic and the total inorganic P in all sediments collected in winter were considerably higher than the summer ones. Okay. However, the total P did not show a clear difference according to the temporal plane. This distribution is possible since abiotic and biotic changes derived from the transition between the colder and the warmer seasons are considerable to main factors for this variation, and essential for the more significant mobilization from the less liable P forms by the growing biomass in sediment. So it's very possible to obtain this kind of results. To see what is the relative contribution of P fraction in order to the total P content, we obtained that the organic P was representing close to the 74 to the 84% in this range in sediments from this lake, which could be derived from the aerosol particles highly enriched with in organic phosphorus, as we see in the pictures, from agricultural lands. In this context, also the main organic fractions contributing to this relative contribution were the moderate libel bound to the fulvic acid, and the organic fraction bound to the humic acid fraction, and together represent close to the 46 to 64% of the total phosphorus. So this fraction, what could be the explanation of this? Because these fractions are recognized as biological, controlling the fitness of the moisture, soils, and sediments, with a clear tendency to accumulate in eutrophic lakes, as we have seen in other studies, and are estimated to be evaluated as good indicators of a phosphorus input from agricultural lands also. Seeing the 31 PNMR results, we observe that the orthophosphate, or the percentage of the orthophosphate, were higher in the less than the more anthropogenized sites in these sediments. On the other hand, the more anthropogenized sediments present higher pyrophosphate, phosphomonosterase, and DNA percentage than the less anthropogenic ones. In this context, biogenetic species such as phosphomonosterase and phosphodiesterase and pyrophosphate are typical of anthropogenetic sediment enriched with organic matter and biomass. And in this context, they were well correlated with the observation for P fractionation. On the other hand, on the temporally plane, the values of orthophosphate were higher in winter than summer. However, phosphomonoesterase, DNA, and pyrophosphate were higher in summer than winter, but not so far. Where the difference between both seasons. This was expected to have a similar distribution that we observe in the phosphorus fractionation, but we had the opposite results. This is a question that we have to investigate in deeper to see what indication is better, or how we can correlate the P fractionation with the NMR spectra. According to the PACE activity, we observe also that at the spatial plane, sediments from the more anthropogenic sites exhibit significantly higher levels of both enzymes. This was positively correlated with the P fractions, and NMR spectra, since sites more anthropogenized present higher substrate concentration for PACE activity. Temporally, higher levels of both enzymes were found in the sample of time during summer. So then this elevated PACE activity in summer may be evidence of a biotransformation of moderately and non-labile organic phosphorus fraction on an explanation of why this form are lower in summer according to the P fractionation, but not concluding with the NMR spectra analysis. About the P gens abundance, similar to the P fractionation and PACE activity, the absolute quantification of the total bacterial community and PACE genes show a spatial temporal distribution also. The spatial distribution, absolute abundance of 16S RNA genes and the 3 PACE genes were significantly higher in sediments from the more than the less anthropogenized sampling sites, as we can see there. This observation supports the idea of higher bacterial activities in these sampling sites, and also the seasons, which was previously based in the microbial organic phosphorus and PACE activity values. However, the relative abundance of these genes at the spatial plane did not show a clear spatial differentiation, as we can see here. Okay, being sometimes these values even higher in the less than the more anthropogenized sites, which has been described for aquatic life environments presenting low organic phosphorus levels. So we have to be careful to interpret these results. The temporally plane, we observe that the absolute abundance and also the relative abundance of the PACE genes in almost of the samples were significantly higher in summer than in winter. These findings are consistent with the observation for lakes with a stronger influence of nutrients-rich waters during warmer periods of year, as we could see in Buddy Lake. And concerning to the general distribution of bacterial peace genes, FOC show the absolute highest values than the others peace genes, followed by FOD and FOX. But in this case, FOC was the biggest one, the highest one. Finally, seeing all the results in the PCI visualization, this confirmed the spatiotemporal distribution of each parameter, analyzed parameter, and a clear separation between summer and winter, and the less and more anthropogenesis sample site, as we can see here. We can see here the red one are the from summer and the blue ones are from winter. And also this group is are the samples with less anthropogenized activity, and here are the samples with the more anthropogenized. Okay. In summer, the libel P-forms, the pace activity, and the total bacteria were positively correlated with sediments from the more anthropogenized sample site. This was evidenced by the high microbial activity and the active upward mobilization of phosphorus from the less to the more libel P-forms during this season. On the other hand, these sediments were positively correlated with the moderately non-libel P-form fractions, also with the total inorganic and organic phosphorus, also the total phosphorus, the total carbon and total nitrogen in winter, resulting from that could be resulting from a co-accumulation of a total organic, the total carbon and phosphorus species implies organic matter complexes during the period of low microbial activity. This could also explain this finding. A positive correlation was observed with bacterial P genes in summer from the less anthropogenic activity. From the less anthropogenic genesis sites. However, no positive correlation were observed between this study, the remaining study parameters, and these sediments in winter. There were not observed the same trend. The Pearson correlation analysis, this table, show very high positive correlations among libel, moderately non-libel, the total organic and inorganic fractions, and also the total phosphorus, also the carbon and nitrogen parameters in both seasons. Here we can see this is summer and this is winter. This is possible since colloidal total organic carbon represents the most critical factor controlling the deposition of phosphorus and nitrogen in sediment of shallow lakes. Almost all the P fractions and also the carbon content and nitrogen content were significantly positive correlated with pace activity, as we can see here. Pace activity. With both enzymes, evidencing on increase on the biotransformation from the moderately and not libel organic P forms to the more libel ones. Conversely, the genes, the PACE genes, were negatively correlated with the observed with bacterial P genes and the P fractions, PACE activity in both sampling seasons. So then high biotransformation rate, for example, can explain this as being part of negative feedback over the induction of PACE genes or a product of a growth of a competitive bacterial population for carbonate sources, as has been this seen in soils, for example. In conclusions, In general, the sediments of Woody Lake present significant spatial temporal distribution of P species, base activity, and bacterial base gene abundance. The P fractionations show higher P fractions in sediments from the more than the less anthropogenized sampling sites, being the labeled microbial, moderately labeled inorganic P, the fraction that were higher in summer than in winter, whilst the moderately labeled organic phosphorus and non-labeled fractions were higher in winter than in summer. There we can find a different trend according to the fractions. On the other hand, 31 PNMR analysis show the higher content of orthophosphate in the less than the more anthropogenized sampling sites, being phosphomonosterase and phosphodiesterase, higher in the more than the less anthropogenized site, being this last result, consistently with our finding by the fractionation activity, fractionation analysis. Also, their summer samples present the higher content of phosphomonosterase and phosphodiesterase, but this was not correlated with our fractionation. Base activities and abundance of bacterial base gene were higher in the more anthropogenized sampling sites with higher P levels, especially during summer. This resuming these 2 points. P contents show positive correlation with base activity and bacterial base genes, regardless of winter or summers. Doesn't matter of this. Whereas a negative correlation between bacterial base genes and base activity was found also. Finally, as outlook, this present study provides valuable information for the role of bacterial community involved in phosphorus cycling from this in sediment of this lake. But the future studies are still need to analyze in deep what happened with the bacterial community there and the influencing mechanism on the base activity and bacteria base genes expression, which might be valuable for design new public policies and normative addressing the environmental protection of this unique environment here in our country. By this, contributing to the protection of this also of their environment, such as, for example, Villarrica Lake, that is other environment we are studying now. My knowledge to all the full EMAlab team, also our international cooperation projects that are supporting as well this researching. Thanks very much for your attention.
Okay, thank you very much, Marco, for your nice work. I had 2 questions from Dr. Fumito Maruyama. The first one is, hi, Marco, do you use qPCR to quantify bacteria and or archaea?
No, just bacteria. I didn't measure archaea.
Okay, but also I want to say we have some data for sequencing.
How through the sequencing analysis also, yes, these are results that we are showing this in this time, because this is part of other article. Here I explain in a full view what they are putting in this article, but we have this also these results.
Okay. And second one is, did you check bacterial activity using their RNA?
No, unfortunately, I didn't check this. This is part of other objective, a field objective, and I am expecting to make microcosm experiment, and there I will try to extract RNA to see what about the functionality of the base genes.
Yes, I want to add also we had another project with Villarrica Lake, a bigger lake, very important in the south of Chile lake, and also includes similar study and includes some
Yes,
RNA extraction. That in the future, I think we had to show some data about
we will. Yes,
active bacteria in lake.
we'll have the
Okay,
result from this.
I think we have time. We can continue. We can invite to Dr. Xiao and Kathleen. Are you there? Please, Claudio, can
I'm here.
Yes,
invite.
yes.
Okay, thank you very much. We have some few minutes
Thank you.
to add additional question and to appreciate thank you to your collaboration with this webinar. I had here. I had a question. Maybe you can read, Dr. Xiao. This is from Jacqueline. Thank you very much, Dr. Xiao. Do you have data about the effect of the glyphosate on the total versus active bacteria community? qPCR data?
We want to check the qPCR data. Maybe the next step. Yeah, we just did the high throughput sequencing of the SNUR 16S RNA. So next step, maybe we need to check the qPCR for the bacteria quantification.
Okay, but I don't know. You had another question, Kathleen Xiao, about the work present here. Marcos, I don't know. You had a question.
I have a question to Caitlin. Caitlin, did you measure the EMAlab spectra of your samples?
Yeah. So I think it's the same student that you guys sent samples to a while ago.
Yes,
so Tony's a grad student.
Tony.
Yeah. So he's just got to the point where he actually started doing NMR on this stuff. So he has lots of samples to do. So yeah, the plan is for him to do NMR on all, I think it's like 7 days over the 2 weeks, but it's for the 6 different treatments with the 3 like seawater, freshwater, mesosaline samples, and then also the 3 controls. So yes, the plan is to have NMR data over the course of the experiment. Yeah.
Okay. Do you have a background about what is the better line to measure? For example, to have a good quality in the spectra. For example, I don't know, the content of total phosphorus could be a parameter to select different samples.
Yeah, so that's the issue that Tony's been having, is that he's trying to figure out how to best set up the scan time for these specific environmental samples. So I know he's had some trouble with that, but there was a cryoprobe that was just installed that I think should be able to decrease the scan time. But so obviously the orthophosphate peak that he has, like, I think 2 or 3 spectra that he's done, the orthophosphate is like always fine. It's just trying to get the monoesters and diesters, like those peaks, to resolve well and be able to differentiate it from the background. So I think right now, as he's trying to figure out the best like signal to noise, I guess, way to alleviate that sort of problem. Yeah, so I think I just gave him a bunch of samples to mess with first. But yeah, the thing is now just trying to figure out the best input parameters when he loads the sample. So the other issue that he had was, I want to say he put it in sodium hydroxide, but the issue is that he had it sitting for too long. And so the queue was backed up. And so if the sample was sitting for too long in like the NMR tube before it was run, the spectra would look really weird. It would look really bad. So he essentially found out that he has to like run it immediately as soon as he dissolves it. So that was like the first problem he dealt with. And now he's again trying to deal with resolving the background versus like what's actually the signal.
Nice.
Dr. Marco, I want to ask you a question. I see a lot of interesting results in your study. And is there any fraction of the phosphorus indicating the organic phosphorus because we studied the glyphosate. You know, the glyphosate is a kind of organic phosphorus compound. So, is there any findings in your study to indicate the interactions between the organic phosphorus and the bacterial community?
I think the best indicator is the mother Adderley and the low label fractions bound to the fulvic acid and humic acid. By this method, it is possible to separate both of these fractions. So, for example, we observed that in summer, during summer, these fractions were lower than winter. This correlates with the activity and also with the paste gene expression. So we can infer that the bacteria community was using this substrate to mineralize phosphorus. But we just have a result about phosphorus. For sure, there in the complex with the fulvic and humic acid, there are a lot of other organic molecules such as pesticide. For sure, there we can find a lot of pesticides and other organic phosphorus. Or another organic molecules toxic for the environment.
Thank you.
Okay.
Thank you.
Yes, go ahead.
I have also a question for Dr. Xiao.
Okay.
We are trying to do the qPCR's measure of glyphosate genes here because we are doing this in the project involving China with us in Villarrica Lake. There we will not what we will find. Because we are extracting sample from 5 different points, but there for sure some sites are low in organic materia. So what is the best, for example, we are not measuring this by HPLC, but what is the best technique or method to assess, for example, a big value on glyphosate, or avoiding the other signals, for example, in HPLC.
You mean how to collect the samples?
No. How you can improve
What kind of method?
Yes. How you can improve your measuring of in HPLC to trying to see what about the bacteria there.
HPLC. We
Yes.
use HPLC to determine the glyphosate concentration.
We will try to use HPLC, but we are only trying to measure activity of bacteria by qPCR. We will just see how these bacteria, for example, we are trying to isolate bacteria, but many of these bacteria should to have maybe not the activity hydrolyzing glyphosate. So many of these bacteria for sure are not has not the enzyme. So what could be the first attempt to see in which place or what place is better to isolation? Maybe
You mean how to we? You mean how we select the typical sediments for the bacteria for checking the yes. We throughout the whole lake we collected almost 60 samples, and we determine the glyphosate concentration. Then we selected from the samples according to the glyphosate concentration, according to the concentration level. So according to the the glyphosate concentration, we find more than 2 milligram per kilogram is a high level, can classified into the high level, and ranged among 0.5 to 2 milligram per kilogram, classified to the median level, the median glyphosate accumulation level, and less than 0.5 milligram per kilogram. We classify them into the lower glyphosate accumulation level. And according to the soil environmental quality threshold of China, the guideline give the threshold just 0.5 milligram per kilogram. So that means that 0.5 milligram per kilogram in the government aspect, they think the 0.5 milligram per kilogram just the threshold, the limitation. But in Baiyangdian Lake, in our sediment samples, we find the 0.5 milligram per kilogram just could be classified into median level. So there may be the serious glyphosate accumulation in this lake. Just since the lake sediment are always considered as the sink of the pollution of the pollutants, and also in the most studied glyphosate polluted area, the Pampas region, agricultural region in Argentina, they find the glyphosate concentration also just ranged in the just 5 or 6 milligram per kilogram. They just take the 6, the 5, or the 7 milligram kilogram at the high level. So and in several publications we checked they just regarded the Pampas as the most polluted by the glyphosate at high level all over the world. They compared European, American, Japanese, the Asia, European, and North American, South American, and they find the Pampas could be
using
that.
glyphosate.
Yes. And we compared the Baiyangdian data with the Pampas, and we find the data is similar. It's similar in a similar level. So we just thought the Baiyangdian Lake maybe has serious glyphosate accumulation, despite the detection frequency is not as high as that in the Pampas region, because we just find near 50% or 20% samples just find the detectable just above 0.001 milligram per kilogram. Yes, the detection frequency is not that high, but the concentration is high. It's as high as that in the Pampas, but the Pampas has higher detection frequency than the Baiyangdian Lake. Yeah.
Okay. Even if there in a place is not a higher concentration of glyphosate, we can find the catabolic bacteria. It is a conclusion maybe, because maybe the method is not detecting the level, but the bacteria for sure will be there. So this could be something interesting to do as well.
Yes.
Are you trying the isolation of a bacteria with the catabolic activity?
We want to, but we are lacking of the students. I have just one student here, and now I join this and listening to this conference now, and he say hi. Come here, come here to say hi to Dr. Miyoko and. And come here.
Okay.
Yes. Yes, here.
Don't worry. Don't be shy.
Okay.
Don't be
Yeah,
shy.
yes.
Hi.
I just want
Hello.
well, and in Fuzhou University, I have just I and him to join with the colleagues in the Beijing Normal University, and together we go to the Baiyangdian Lake to collect samples.
Okay.
And after taking back the samples, I and Kwangbo just two people to analyze. So a lot of work we need to do, but we are the liver is lacking.
Yeah.
So we can share our experience with you because we are trying to do this in a near future, isolating glyphosate
Yeah.
catabolic bacteria
Thank you.
and also atrazine. Are you trying other pesticide or just glyphosate?
Another pesticide.
Just in recent days, in just a short period, we just focus.
Okay.
We want to focus on the glyphosate.
Yes.
Yeah.
Yes. Nice.
Yeah, because Dr. Miyoko gave us the proposal and write the herbicide such as glyphosate and then the other. We just choose the first one, glyphosate, and then we determine the concentration, and we find
Yes.
the concentration level is high. Yeah.
Yes.
Yeah, at least where I am, glyphosate is definitely one of the top 3 herbicides that is used. I just don't know off the top of my head what other ones, but definitely glyphosate is like used everywhere.
What news all around the world?
Yeah.
Before finish, the last question is related to glyphosate. Do you know some alternative to replace glyphosate and as an effective alternative in terms of health, environmental, and economic aspect? What is the situation in China? I think it's about glyphosate.
We just go to the agricultural chemical stores to check. There are many.
No, disconnected.
Oh no.
No, again.
Okay.
Oh, maybe she's back? Okay.
Yes. Oh, we can wait for a few minutes.
Caitlin, did you?
The screen is black. Okay.
No.
And we find that there are many agricultural chemicals sold in the stores. There are many similar herbicides as glyphosate were selling in the stores. They are more toxic. And more active, more efficiency than the glyphosate.
Okay. I think we had on time.
Can you hear me?
Yes,
for sure.
Okay.
I think we are on time because now here 11:30 and China is late. Yes, please go ahead.
It's my pleasure to talk with you and learn from you. Hope you can give us very important information and suggestion, and we can learn from all of you. Yes. Thank you for your invitation. Yes, we can finish.
No, we hope before finish we present closing video for the transmission, but we can internally keep connected to talk. Okay.
Yeah.